Thank you, Luke, for introducing me. I finished the PhD in National Institute of Mental Health, Czech Republic, Prague. And my PhD was about animal models of psychosis using different psychedelics. So I will show you part of my results. And I would like to talk shortly, briefly about the history of Czech psychedelic research in my country. Then I will go to antidepressant effect of Psilocybin and underlying neurobiology with special emphasis on neuroplasticity. Okay, so first, let me introduce the psychedelic research in my country. We use, during the 60s and 70s, we use different kind of approaches: psychomimetic approach, psychedelic approach, experimental approach, and psycholytic and entheogenic approach. The main personalities of psychedelic research during this era were Milan Hausner and Stan Grof, which both of them started in Czech Republic in the same lab as I started to work a few years ago. And here you can see Dr. Hausner in his experimental room in Satská clinic. He treated approximately 3,000 patients during with LSD in this place. And because of the huge need of LSD in Czech Republic during this era, we started to manufacture our own LSD in Spofa factory that is near Opava, the city on the north of my country. There were main indications of psychedelic research, but part of them were also anxiety disorders and depression, which is important for my presentation. Czech people are really interested in psychedelic mushrooms. I've been picking mushrooms since I was 17, and the reason is probably that Czechs are a mycophilic nation due to Gordon Wasson, and we have very potent mushrooms. We harvest Psilocybe semilanceata on the meadows, and we harvest Psilocybe bohemica and Psilocybe cyanescens in forests. After almost half a century, psychedelic research started again in Czech Republic with the study of my supervisor, Tomáš Páleníček, and we started to administer psilocybin approved for human use in a clinical trial, which was double-blinded and placebo-controlled. We used 20 volunteers, mean age 35.6 years. We all university educated, and we used the dose of psilocybin from 17 to 24 milligrams in order to model psychosis. We had 7 from the 20 volunteers were naive to psychedelics, and we had 1 female non-responder. We followed the guidelines for hallucinogen use, and here you can see our setting. I will show you some results shortly from this study. Of course, we observed a huge psychedelic effect of psilocybin measured by altered states of consciousness scale. Psilocybin increased anxious ego dissolution, oceanic boundlessness, and visual reconstruction very significantly. Importantly, we didn't observe any clinical relevant side effects. All subjects were monitored 1 day after the session and the 3rd day after the session. And during the session, they exert only mild sympathomimetic effect, increased systolic and diastolic blood pressure compared to placebo, and increased the heart rate. And another important thing is that we observed positive long-term changes after 1 month using persisting effect scale. We adopted mainly the psychomimetic approach. On this slide, you can see the comparison of animal data of EEG and human data. This is the mean power of the EEG in rats and in humans, and you can see that these curves are quite similar in the shape. Psilocybin leads to decrease in dominant activity of rat, that is beta, and the decrease of dominant activity in humans is alpha, and the increase of the rest in higher frequencies. Then we assess the functional connectivity using the EEG coherences, and we found that psilocybin in rats decreased EEG coherences in lower frequency bands, both in humans and in rats, and the most significant decreases of EEG connectivity were in frontotemporal areas, which reflects the finding in acute psychosis. In humans, we observed increased coherences in higher frequencies. I want to another outcome from the study was the trip reports from the volunteers, and I would like to show you the feedback after 1 year of our volunteer, pointing to flexible thinking and creativity. The experiment has a positive influence on my experiencing and my overall satisfaction in life. It occurs to me that on my normal days, my thinking is constrained to a narrow corridor of hardwired patterns, that I am like a leaf floating in a wild river, tossed amid the rocks by the stream, feeling that I am at the mercy of life for better or worse. During the psychedelic experience, the mind is liberated from stereotypical patterns. It looks for interpretation and examines options how to handle life, bringing spontaneous and original insights. Although some of these ideas are later often dismissed as unfeasible by the sober mind, the uplifting effects of the very experience of unlimited freedom, thought and creativity are still present long after the intoxication has worn off. So even though I don't have so many ideas anymore, I know that such a thing is possible. I know my problems are relative and there are plenty of solutions, several of which are always positive. This was after 90 milligrams. And here you can see again our experimental room, which in fact is a Faraday cage because of the EEG signal. And now I want to answer the question why our volunteers are motivated to undergo the experimental intoxication. We administered to potential volunteers online survey about the demography, about the drug use and the motivation for going to the study. And we used the sample only volunteers which previously contacted us, our research team, by email. The sample represents highly motivated individuals who wanted to undergo the experiences in controlled conditions. Our sample was 143 volunteers, mean age 35.8, and most of them were university educated, and most of them were males. 63 of them has previous psychedelic experience, and importantly, 37% of them were psychedelic naive. So they want to have their first experience in experimental conditions, controlled conditions. Here you can see the drug abuse of our volunteers. The black columns represent the repeated use, which is defined at least once per week for the period of 1 year. And we found that 40% of our subjects did not use alcohol regularly, which is quite rare in Czech Republic because almost all people in Czech Republic meet the criteria for repeated use of alcohol. Subjects were not used psychostimulants and opiates regularly, and 7% of them used regularly benzodiazepines. That's probably because part of them were patients suffering from anxiety. And what is really important that they didn't meet the criteria for repeated use of hallucinogens. That points to that hallucinogens are not addictive. And we look more specifically of lifelong use of hallucinogens in our potential volunteers. We use a made-up drug called Relevin, exclusion criteria. So they take the Relevin, their answers are completely excluded because Relevin doesn't exist. Then we found that Psilocybin was used mostly experimentally in our sample. Almost 50% have ever tried Psilocybin, but only 2% of them has more than 20 experiences. Very low proportion of our sample used any psychedelic drugs more than 20 times. That means this sample were not real experienced psychonauts, but more people who really want to have this experience in controlled condition because they are not very used to in psychedelics. Most of them repeated use was MDMA, 1.5 has more than 100 experiences. Our volunteers expected from the participation in research self-exploration more than just adventure, and they want to advance the research, which points to some kind of altruism or strong belief in psychedelics in this sample. There were a low rate of potential patients, probably because of our study was mainly for or only for healthy volunteers, but they didn't know that. They tried to participate and to solve their psychiatric conditions. And these conditions were mainly anxiety, depression, or a combination of anxiety and depression. So this leads us to the question, do we have some evidence that psilocybin could be useful for therapy of depression? Here are some relevant studies. Roland Griffiths found that deep internal and spiritual meaning of psychedelic experience were 70% rate experience among the most 5 meaningful experiences in their life. Studios found that 40% of respondents of their experimental studies in Switzerland report long-term positive changes. Catherine McLean found the increase of the trait openness, like a personality trait, after psilocybin intoxication. Grob found decreased anxiety in patients suffering from cancer, and Gesser says similar finding with LSD. Sanchez found the antidepressant effect of ayahuasca. And of course, Robin's group published the first evidence that psilocybin is effective for treatment of depression, which was presented in our last presentation. What are the mechanisms of action? Psilocybin is a serotonin agonist of 1A, 2A, and 2C receptor. So it seems that it can have something in common with used antidepressants. There are two approaches how we could treat depression with psilocybin. The first one is small repeated doses called microdosing, and there are reports of increased creativity, improved focus, enhanced perception of sensations. And the second approach is a single dose with which can trigger transformational experiences. And we think that these experiences are mirrored in a brain structure or function by neuroplasticity and connectivity. So what are the biological and psychological variables underlying the antidepressant effects? From the psychological point of view, we can distinguish the dynamics of psychedelic experience. After drug intake, there is a psychedelic state depending on a substance which lasted from minutes to hours. And after that, there's an afterglow effect, days to weeks or 1 month after the intoxication. And there is some residual effects later. During the psychedelic state, one can reach the peak experiences that is experienced beyond everything that individual experienced so far. The degree of antidepressant effects correlates with the depth of psychedelic experience. The afterglow period is characterized by elevated mood energy, decreased anxiety, guilt, and increased capacity for close interpersonal relationships. This was confirmed by our study as well, because during the phone calls, 1 and the 3rd day after the study, our volunteers reports this afterglow effect. We assume that during the afterglow there is a increased effect of psychotherapeutic intervention. And what are the biological mechanisms underlying the antidepressive effect of psilocybin? So psilocybin, as I told you, is an agonist of serotonergic receptors, and on this schema I will show you the first 3 levels of the effect of psilocybin. On the first level is the inhibition of dorsal-radial nucleus, which is in the brainstem, and there is a direct presynaptic inhibition through 1A receptors and indirect inhibition through stimulation of 2A receptors on GABAergic interneurons. This level. The second level is a thalamic inhibition due to stimulation of 2A serotonergic receptors on GABAergic interneurons there's a decrease in the filtration function of the thalamus, which leads to sensory overload of the cortex. And on the third level, it is increased of excitability due to agonism of 5-HT2A receptors or glutamatergic excitatory pyramidal neurons. We believe there is a link between neurobiology and the phenomenology of the intoxication. So here, this is the localization of the alpha decrement observed in our study, and we localized the decrease of the alpha power using chloreta, that is low-resolution electromagnetic tomography, in the posterior and midline structure of the brain. So we replicate the results from the group around Robin, and we expect that this will correlate to ego dissolution. We observed increased activity in higher frequency band, beta and gamma, in a broad associative cortical areas, and we expect that this can lead to like unifying effect observed as a synesthesia or a feeling of unity. The frontotemporal disconnection is observed during acute psychosis as well, and here we think that it's a correlate of the increased entropy, chaos, and divergent thinking potentially with creativity. So this all leads to points to therapeutic and self-developmental potential of psilocybin. Recent findings from neuroimaging studies confirm this as well, and all of them pointing to unpredictable neuronal activity and disorganization of neural networks. We used more detailed approach to go deeper into connectivity using Iloreta connectivity lect coherences, and here we observe that in the first 3 hours there are mostly decrease of EEG lect functional connectivity in alpha band and increases in delta and higher frequency bands. And this pattern completely switched after 6 hours into decrease of delta and increase of higher frequencies. So I think this could be one possible, or not explanation, but a link to the explanation of the long-term effects, like rewiring of the brain. There are two main hypotheses about the entropy and neuroplasticity. The entropic brain hypothesis assume that we can treat the rigid state through transient increase of the entropy of the brain, and then the long-term increase of entropy. And there is a self-optimization hypothesis from Tom Fraser, which says that if we increase the transient entropy, then we can establish a new balance in a brain, which exerts decreased entropy more than the previous state. Both of these transient increase of entropy can trigger neuroplasticity. I think that similar things happening during the electroconvulsive therapy, for example, because it's increase of the brain entropy due to brain stimulation as well. And we know that ECT stimulates, for example, brain-derivated neurotrophic factor increase of BDNF, which is responsible for the antidepressant effect. Similar thing can be like a physiological sleep-wake cycle, because during the REM sleep, the entropy increase every night and then decrease during the non-REM sleep. We can divide neuroplasticity processes into neurogenesis, which is emergence of the new cells, mainly in hippocampus, and into synaptogenesis, which is a functional remodeling of neuronal networks. 5-HT2A receptor can be responsible for these changes because its hallucinogenic effects are dependent on this receptor. Its most abundant serotonin receptor subtype in the cortex is targeted for hallucinogens, but also for antipsychotics, anxiolytics, antidepressants. It's associated with several psychiatric conditions that are associated with aberrations of dendritic spine morphogenesis. We have some evidence that psychedelics increased neuroplasticity in vivo. here is hippocampal neurogenesis, and this is synaptogenesis. Ketamine increase the hippocampal neurogenesis, increase BDNF, and increase expression of neuroplastic genes in frontal cortex. LSD increase the genes in neuroplastic cortex, and DMA, there is a study both increased neurogenesis and neurotrophin 4. And psilocybin, there is one study where was the low dose of psilocybin stimulate the hippocampal neurogenesis. However, the higher doses decrease the hippocampal neurogenesis. What is quite important, I think, is one study, recent study of ayahuasca, where was observed that in chronic users of ayahuasca, using ayahuasca more than 100 times, there were structural changes in the brain. In more detail, they observed the decrease of volume of posterior cingulate and non-significant increase in anterior cingulate. This can be linked to remodeling of the brain under psychedelics. We have also some proofs that psychedelics induce neuroplasticity in vitro. The OY induces spine remodeling after 30 minutes in neuronal cultures, and DMT, which can be the endogenous substance in our brain, because it was proven that it's contained in the rat brain in 2013, has a neuroprotective effect during the hypoxia in neuronal cells. So it increases the survival of the cells exposed to hypoxia. And there are some new results which were presented on Psychedelic Science by Jordi Riba, that DMT has some neuroplastic effects itself. So to conclude, pilot studies show the increased efficacy of psilocybin on treatment of depression. Neurobiology of antidepressant effect can be caused on a receptor level by 2A agonism, on the neural level, on the network level, acute disorganization of neuronal networks and increased entropy, and long-term effects can be reflected by changes in connectivity and neuroplasticity. I think there is a very important role of set and setting, and we need further research to go deeper into the extra pharmacological variables. And there is an important paper from 2016 from Ido Hartogson. And another important thing is effect of psychotherapeutic integration, which is difficult to measure. And I think that this can be very effective during the afterglow period. And therefore, there is a big need of specialized courses for psychedelic psychotherapy for citrus. This is our research team of National Institute of Mental Health. The studies are leaded by Professor Horáček and Thomas Pálinčík. And I would like to thank you for your attention. Thank our 20 healthy volunteers and my colleagues. And at the very last, I want to bring to your attention that we are starting the crowdfunding campaign for psychcare or psychiatric harm reduction festivals in Czech Republic at the moment. Thank you.